The second part of chapter 12, we're going to focus on antimicrobial resistance and some new approaches to treating microbes. The term drug resistance is just referring to some type of adaptive response that an organism has that can tolerate a drug that normally would be inhibitory. So not all bacteria are susceptible to every antibiotic. They're naturally resistant. Here we're talking about the ability to adapt to a drug that it would normally be effective against, but now it's not. And this is due to changes in its genetic versatility and adaptability. So again, there are some intrinsic resistance versus acquired. So intrinsic resistance is just a bacteria that normally is resistant because of something they can produce by themselves, versus acquired, which is where that resistance is, again, the bacteria was resistant or was susceptible to it. It was sensitive. So it was previously sensitive, but now it's resistant. So again, intrinsic, it has like always been resistant to an antibiotic because they can make something or defend themselves. But acquired means they were sensitive and now they're not. So looking at some ways that this resistance can occur, starting with spontaneous mutation, especially in viruses that don't really proofread whenever they're replicating their DNA or RNA. A lot of times they will just spontaneously mutate. They'll add the wrong A, T, C, or G. They'll change the genetic makeup. And there is always a minimal chance that that mutation, instead of making it ineffective, actually provides some type of advantage to it. And then there's even a smaller chance that that mutation will occur, will allow drug resistance. However, even though those chances are slim, because there's such a large population, the constant rate of mutation ensures that eventually the odds will be in the favor of the bacteria. That even though there's a small chance, eventually the mutation will occur that gives them some type of resistance. Now, the more common way in which this happens, and one we worry a little bit more about, is through a process called horizontal transfer. And this is where there's resistance through what's called an R factor or resistance factor. So back to those plasmids, again, the extra pieces of DNA that bacteria have that give some advantage. So if the plasmid is specific to providing resistance to a drug, it's called the resistance factor. Well, that can actually be transferred from one bacteria to another through the process of what's called conjugation, which is using the pilus to transfer, kind of connects the two bacteria, and the plasmid transfers through the pilus. Through a process of transformation, where the bacteria can actually take up just random DNA in its environment, or transduction. And in transduction, they're actually using bacteriophage as a way of transferring bacterial DNA. So these transposable resistance sequences, or what are called transposons, they can also occur. And this is where it's duplicated or inserted from one plasmid to another in the chromosome. And this accounts for, again, why there's so much drug resistance. It's mainly due to this horizontal gene transfer. So these genes are transferred extremely frequently in nature, much more common than mutations. And it can be from unrelated bacteria, between bacteria and viruses, or vice versa, or other ones, especially those living in the normal biota or the environment. Now, some other ways is actually making new enzymes. So these enzymes could inactivate a drug, but this only occurs again with some newly acquired gene that gives them an enzyme or makes a protein that destroys a drug. It can also change the permeability or uptake. So by decreasing that, and this is mainly from a mutation that changes their receptors, or it changes the permeability. So now they're no longer being taken into the cell. And then finally, by elimination. So whenever the drug comes in, there's something newly acquired that allows it to pump it out. Okay, so again, if we kind of look over here, again, penicillinase, for example, is an enzyme that inactivates penicillin. Here, looking at the permeability, So normally the receptor attaches to the drug and it brings the drug in. However, if there's a mutation to that receptor, now the drug doesn't bind to it, so it can't get in. Or again, another option is that the bacteria could produce some type of pump or transporter that pumps the drug out. Another way is that the binding site for the drug either decreases or it changes its affinity or ability to bind. And these can occur either by mutation or acquiring new genes. And they can affect the metabolic pathway to shut it down, or it can use another alternative pathway that gets around the drug. And those are due to mutations of the original enzyme or enzymes. So for example, here with the binding site, so normally this drug would bind here to the large ribosomal subunit and prevent it from working. However, due to either, again, acquiring a new gene or a mutation, that protein or that ribosome changes a little bit, and now that drug cannot bind. So therefore, it's ineffective and it keeps going through protein synthesis even in the presence of the drug. Or the other way, by affecting the metabolic activity. So here, normally the drug works on this enzyme that converts one molecule to another. Here, the bacteria, either through mutation or acquiring new traits, is able to go through a slightly different pathway and still make the product. Now, natural selection then is what allows drug resistance to continue. So anytime you have a large population of microbes, again, the idea of natural selection is that there's survival of the fittest. So a few of those individual cells will naturally be resistant to a drug. So if the drug is not present in the population, then the bacteria that have resistance are kept low because everyone's just kind of competing and surviving the way that they're supposed to. However, if you have a few that are resistant to a drug and then you expose that population to a drug, now what happens is you get an imbalance. All the sensitive microbes are going to be killed, and now that allows all the resistant ones to become the largest part of the population. And then they're the ones most producing, most prevalent in the population, and that allows it to proliferate. So now the offspring of those are resistant. They're more likely to replace the population with mainly drug-resistant forms. And so eventually, the whole population, due to that transferring of information, is completely resistant to the drug. And that's what this figure is just showing. So again, normally we have a large population and very few are resistant to a drug. However, if you add the drug and now all of these guys get killed, and now you just have mainly the two resistant bacteria around, now that allows them to mass produce and become overpopulated. And eventually, even if you have a few that are sensitive to the drug, eventually, due to that horizontal transfer, they're going to allow that non-resistant drug to become resistant. And then, again, eventually, the whole population is now resistant to the drug. So what is the role that we play? So unfortunately, 75% of antimicrobial prescriptions that are used for common things like sinuses, upper respiratory infections like colds and that kind of thing are likely viral, and they don't benefit from antibacterial drugs. However, unfortunately, most people, when they go to the doctor for these types of with these types of symptoms, a lot of times they're prescribed an antibiotic. So they're giving kind of this shotgun approach that even though they don't know if it's bacterial, they're like, oh, let's just give them an antibiotic and see if it helps. But that's just increasing. So we have this excessive use of antimicrobial drugs that are made and used in the US as well as sent to other countries. Now the hospital plays a role too, because it continually exposes pathogens to drugs. I mean, that's what they're there for, is to treat drugs. However, hospital houses these susceptible patients that already have a weakened defense. So the workforce may not strictly adhere to all of the precautions, especially today when there's shortfalls and there are, healthcare workers are kind of overworked, there aren't enough of them. And so, things kind of slide or they're not caught. So this has actually led to penicillin resistance and nearly about 100% of all Staph aureus strains in the last 30 years is due to overuse in the hospitals. Another issue is using drugs in animal feed. So about 80% of all antibiotics are actually given to livestock. This allegedly decreases infection. Again, it's thought to be prophylactic, preventing the infection to improve the animal size and health. However, they're enteric, so the bacteria inside share those resistant plasmids with others, and it just increases that drug resistance. So the pathogens jump then to humans and share those drug-resistant genes that continue to cause problems. Unfortunately, we're really at a very urgent problem where it's a real possibility that we're about to enter a post-antibiotic era where some infections will become untreatable, especially Staph aureus. We already have MRSA and VRSA where they're resistant to a lot of antibiotics. It's very likely they will continue to develop resistance until we don't have an antibiotic to treat them. So the CDC has actually categorized resistant bacteria into 3 groups. So we have urgent threats, serious, and concerning threats. So they are constantly looking at the amount of antibiotic resistance in different microbes and kind of classifying them in one of these 3 categories. So now we have the bad news. Let's talk about some of the good news, some new approaches then to antimicrobial therapy. So our kind of past approach has just been to find new drugs for known cell targets, kind of custom designing drugs to those targets. Again, kind of the same idea, just modifying them a little bit. However, now we have some different approaches like nanomaterials, what's called antisense RNAs, CRISPR technology, actually using those bacteriophages, the viruses that infect bacteria, and then antibiotics specifically targeting critical gram-negative proteins on the outer membrane. In addition, kind of helping nature along, we can use these probiotics. So these are actually live microorganisms that are fed to animals and humans to improve the intestinal biota. So this helps to replace lost microbes, especially while under antibiotic treatment. So again, it prevents those super infections from occurring. So it keeps the good bacteria present and also supplement the biota that's already there. And these have been shown to be very safe and effective. Then a prebiotic. So these are actually nutrients. So it's not actually the whole organism of good bacteria. Instead, it's nutrients that encourage the beneficial microbes in the intestine. So they're essentially feeding those good bacteria. And then there's even fecal transplants, which I know sounds absolutely horrible, but it's a treatment that can be used for that main superinfection we talked about, the antibiotic colitis, where the Clostridium difficile infections occur. So what this does is it actually transfers feces that have the beneficial normal biota from healthy patients via colonoscopy. So it inserts those healthy bacteria into those under antibiotic care to kind of reestablish the normal biota. So again, lots of kind of new approaches that are out there and some other things that can kind of help our normal microbiome to prevent those super infections from occurring.